first I want to thank Klaus and the Royal Swedish Academy for the honor, the Crafoord family for recognizing the need to do these sorts of things, and the recognition across all sorts of disciplines. I think it's fantastic that they do this, and I'm humbled by the award, and I'm honored to be with my distinguished colleagues in this particular phase. This is going to be a little bit different from the previous two talks. This is going to be more of a historical narrative in some respects about how science has been done and lessons we can learn from it. And hopefully it'll be interesting, and if not, well, we'll try to be on time so you can get to lunch. As noted, I had a slightly different historical scientific background, and I'll come back to why that was the case, but I've been at biotech companies for most of my career since I got out of my postdoc. And it's largely been with the same group of people for about 15 of those years. And one of the things we did when we started was we tried to figure out what projects we should work on. We wanted to work on autoimmune disease. It was really important. I fell in love with immunology in college, and it touches so many aspects of our biology that I felt like it was an important thing to work on. So one of the collaborations that we established was with Oak Ridge National Laboratories. So this is in rural Tennessee. You'll notice it's rural. This is the laboratories. It's surrounded by a lot of trees. That, in fact, is by design. Oak Ridge National Labs was built basically in a year and a half in the early 1940s as part of the Manhattan Project by the Department of Defense. And it was a secretive facility. People didn't know where it was. There was no Google at the time, so you couldn't do Google Maps. And it was hidden away on purpose because what was done there was work on principles of radiation development, and including the generation of nuclear weapons. So again, part of the Manhattan Project. It's difficult to read the sign, but one of the things that you would see when you went into Oak Ridge National Labs is, this says here, is what you see here, what you do here, what you hear here, when you leave here, it stays here. So it's a very secretive facility. People didn't know what was going on, other than the people who worked there, of course, but what was going on was people generating nuclear reactors and other things to support the war effort. What they also did, which not quite as many people know about, is they developed a mammalian genetics unit. And it was headed by two people, Leanne Russell and her husband, Bill Russell, both members of the National Academy of Sciences, outstanding geneticists. And what they did was they built a mouse house, and you can see the mouse cages here. And it became one of the largest mouse houses in the United States during the course of the war. And their goal was to understand what effect radiation and other mutagens would have on genes, traits. And this was done very early on. So you can see the old-style mouse cages here, et cetera. You can see Lee has a very nice, friendly mouse. They're not all that friendly. From a timeline perspective, this was started around 1943, right around the time the Manhattan Project was started. World War II ended in 1945. We didn't know the DNA structure until 1953. We weren't even sure that nucleic acids, DNA, was in fact genes until the early 1950s. So this was really begun before we really knew that DNA actually coded for the genes and the traits. But the government and lots of other people wanted to understand what the effect of radiation was going to be on traits. And so they established this mouse colony, and Lee and Bill Russell they would do many different things, but one of their attempts to figure this out was to gather up a whole bunch of mice, put them in the back of their Volvo, drive to White Sands, New Mexico, and put the mice at various distances from a test bomb that would go off. They'd go into a bomb shelter, test bomb would go off, get up, collect all the mice, take them back to New Mexico, and see in their offspring how many mutations they had. And by doing that, they actually got the first sense of what occupational health safety would be in terms of how much radiation you could be exposed to. They were some of the first people to suggest that pregnant women should not get x-rays. And so again, this is very early days in the process of all of this, and radiation was very new as a science. The Scurfy mouse, which is what I'm going to talk about, and both Sasha and Shimon have alluded to this, was actually a spontaneous mutation that never actually was exposed to any mutagen whatsoever. Probably the most famous mouse to come out of this facility, and it was never mutagenized and was never actually part of the program, which I find incredibly ironic. It was found in 1949, and a little bit happened between 1949 and 1998, but not a lot. And there's a couple of things I'll point out. Things like the Cold War ended, but other things around this mouse, very little happened. As you heard from Shimon, he really identified these regulatory T cells back in 1995, resurrecting the suppressor field. As was mentioned, the suppressor field disappeared in the mid-'80s, and I was one of those people who left it behind because in the middle of my graduate school studies, it was proven at the time that suppressor cells couldn't possibly exist, and so everyone, not everyone, Shimon not, but many of us abandoned the field. And a great lesson in this is if you can stick with it, sometimes it really pays off. And so now today, these are used in clinical trials. Also note that the human genome sequence didn't really appear until 2001. So again, everything that happened in this particular space was well before we knew what the human genome looked like. So the mouse itself, heard a little bit about this already. You can also see that graphics illustrators back in the 40s were a little bit different. And they described different types of mice. So this is a spontaneous mutation, as I mentioned. It was recessive and it's X-linked. That means half the boys of any carrying female would come down with disease, and it was lethal. 3 weeks after birth, these mice died of massive inflammation everywhere. So it was recessive, sort of like this mouse with the martini sitting by himself at the bar. It was also sex-linked. This is the first sex-linked gene that was ever identified in the mouse. And in fact, one of the major uses of this mouse before associating it with regulatory T cells was that it proved the Y chromosome conferred maleness. People didn't really understand whether it was lack of two X's or whether it was a Y or what the role was. This proved that the Y chromosome conferred maleness. So one of the things that I think is really amazing is that Bill and Lee Russell knew that this was an important mutation. They knew that the phenotype was dramatic and it was incredibly penetrant, and it was just very important. So they kept this strain alive from 1949 to the mid-90s, which meant they had to breed a mouse, look at the offspring of that mouse, look at the males, ask whether or not any of the males had the disease. If the answer was yes, they would take that female and look at her females and breed them again. And so they had to do this iterative process to keep this mouse going because they didn't know what the gene was. So they kept that alive for over 300 generations of just continuous breeding, which was pretty amazing, actually. And in the late 80s, early 90s, a couple of people, Virginia Godfrey and Irby Wilkinson, really showed that this was immunologically relevant. Irby did experiments in which he put a single T cell receptor into the mouse, and if you did that, if you made the T cell only see one thing, and that thing turned out to be chicken ovalbumin, the mouse did not get sick. And so that says whatever those T cells see in that mouse, it had to be part of the mouse structure. So the question becomes, why did we clone this gene? And I got into this field because I love immunology, but also because I wanted to actually find therapies for things like rheumatoid arthritis, polyarthritis, other autoimmune diseases. I think we really wanted to understand enough about the biology to treat this in an intelligent way. And so for arthritis, there are fairly good therapies now, but there's still lots of autoimmune diseases, and as people have described, the incidence is simply going up. But most people are perfectly healthy. This little girl eating her pizza is perfectly healthy, and she has enough cells in her, enough immune cells in her to see anything. So I should describe that. Individual cells can see virtually anything that nature can throw at her. Any pathogen, any worm, any virus, any bacteria, she has the cells that can see that. We vaccinate people so that we have more of them and they can respond more efficiently and more effectively. But we're actually simply boosting an existing response. And so I really wanted to understand why she was so normal and what goes wrong in patients who have polyarthritis. And really it's about this balance. So the immune system, when it's working right, gives us health, protects us from pathogens and cancer as well. And when it's not in balance, we get autoimmune disease, we get asthma, we get a whole variety of other conditions. And the Scurfy mouse was a dramatic manifestation of this side of the equation. And it doesn't really matter how you look at this, but any way you stimulated the cells from this animal, they were hyper-responsive. They were beyond anything you'd ever see in a normal animal. And so that told us that the T cells themselves were intrinsically altered. So again, this was back in the mid-90s. We didn't have the human genome sequence. We knew this was on the X chromosome, but it was on a fairly large chunk of the X chromosome, 3.33-ish megabases, and that was simply too big to sequence. So we had to actually compress the region so that we could be able to identify the genes that might be responsible for this. And this was done by a tremendous geneticist and colleague, Mary Brunko. We were a small biotech company. We didn't have a lot of resources. We didn't have a mouse room, which made this very difficult. So we actually took over a janitorial closet in our building. And so just kind of shows the level of initiative you need to go to when you really want to get something done. We did this for two reasons. One, we didn't think we could make it smell any worse with the mice because it already smelled terrible. And the second reason was it had the only sink in the building large enough to wash the mouse cages. So we generated a lot of mice in this janitorial closet over the course of about 6 months so that we could shrink this region. And it worked. We got down to 20 genes. We got down to this size region. We got down to 20 genes, which we had to prioritize. And the only way to figure out the mutation at this point was to sequence each one of these genes from the normal animal that was perfectly healthy and the mutant animal that was sick. And we had to hope that there was going to be a difference in some amino acid sequence between, or some nucleotide sequence between those two strains. And that was one strategy to do this, and it was the strategy we took. And so we prioritized these, and I got to do the final prioritization of the 20. And I was pretty confident the top one was going to be the right gene, because just based on its characterizations. And I know you can't read this, but a lot of these things are just numbers, because we knew they were genes, but we had no idea what they did. They just were never described in the literature. People didn't know about them, and this was the first time anyone had seen them. So we prioritized this. We sequenced these. It took us, I think it was 3 months. It might have been longer in 1998. Today, this would take less than 3 hours to do, given the technology. It's pretty impressive. So we sequenced all the genes. And as you might guess, my intuition is terrible. The gene that was responsible is actually the 20th gene. It was the last gene we sequenced. And so in all of this, I think you have to get lucky. This was not an example of getting great luck. It would have been nice if it was one of the top 5, but I was still thrilled when we got the gene, except for one problem, which is that the reason it was on the bottom of the list was twofold. One, it was, by looking at expression, found in every tissue in the body. Kidney, liver, heart, it didn't matter. They expressed a little bit of this gene. Well, that didn't make any sense with the phenotype, in my opinion. I thought, well, it can't be that. That's silly. And the other reason was it was a transcription factor. That is, it bound to DNA and turned on or off other genes. And I knew nothing about transcription factors, and I did not want to study them. But genetics is genetics. Genetics tells you truth. So this really was the problem. This really was the problem in this mouse. And so you have to follow the data. You have to follow what's actually true. And so this is just a little diagram of what that looks like. The mutation actually was 2 nucleotides in the gene sequence. So this was this 500 kilobase region where we sequenced each one of these little genes. The FoxP3 gene sits just about in the middle. There's a 2 nucleotides that get stuck in the middle. And what that does is it throws the reading frame off. And so downstream it stops. It's like putting a random set of letters in the middle of a sentence, and suddenly your sentence doesn't make any sense anymore. And that's what happens. And so it turns out that just stopped the gene from being, or stopped the protein from being produced, and so we knew that this mouse would make no protein. So that was great. We knew what was going on. This is roughly what it looks like. It's called a winged helix protein. So this is the double-stranded DNA that Watson and Crick solved for us. This is a helix, this is a wing. This is a wing. The thing in orange is FoxP3. This is a helix that sits down in the groove of this DNA. It's not quite this simple because this thing usually binds to a number of other transcription factors in a complex, but just for simplicity's sake, this is more or less what this looks like at the structural level. So we were pretty confident we had the right gene, but genetically, you have to prove that. And so to do that, you have to put the normal gene back into the mutant animal. And so we did that by making a transgenic mouse. And this was one of the better days in my scientific career. Mary and I decided we had to be the people who looked at this because we were too invested in this. So this is a lymph node from a normal mouse. It's, I don't know, that big. I don't know how many cubic millimeters that is. This is a lymph node from a mutant mouse. It's much larger. It's full of cells. They're all activated. They're all making some of the cytokines that you've heard about. And this is a mutant mouse that also carries a normal FoxP3 gene. And so it looks just like the normal mouse. It's actually a little bit smaller, but that's okay. And we made 5 different lines of these, and they all looked the same. So when we saw that, we knew we had the right gene for sure. We had genetic confirmation. We were ecstatic. We had a big party, actually. We also called collaborators at the University of Washington. You've heard about IPEX and the University of Oregon Health Sciences. You've heard about IPEX, again, a rare X-linked disease in humans. This is one of the families. It was discovered in 1982, and there are not a lot of families in the world. I think there's about 200 to 300 known families in the world who carry mutations in this particular gene and have this particular phenotype. But this is what the family looks like, and you can see male children here in squares are quite often not surviving. So an example right here is in this pedigree, a carrier female and her husband have 2 children, 2 boys, 1 survives, 1 has disease. And these children generally don't live past 3 years of age without really intensive therapy. These are mutations that are now found in humans in IPEX, and so there's about 75 different mutations that cover those 300 families. Almost all of these end up destroying the protein in one sense or another. There's probably more, but to date, I think that's kind of where we are. So when we saw this, we knew it was obviously really important. We knew it's immunological. We wanted to understand a little bit more about the mechanism. And like Sasha, I like to go back to the old literature and read what some of the earlier scientists have said, because they tend to be right quite often. And so Paul Ehrlich, who won the Nobel Prize in 1908 for his studies in immunity, described this process, which says the organism has certain contrivances to control the immunity reaction because it's too powerful. And so he recognized this back in 1901. He referred to it as the horror autotoxicus. That is, it would be you reacting against yourself and destroying yourself, which is exactly what Scurfy mice and IPEX patients look like. And one way to think about this is if it's a rheostat or volume gauge, temperature gauge. So if you have no copies of the FoxP3 gene, whatever stimulus you give, the response is very dramatic. If you have 1 copy, normal, you get a normal response. If you have more than 1 copy, you get a blunted response. And that number is very proportional to, the response is very proportional to that number. So if you have a lot of copies, the response is really, really bad. And if you have 5 copies, it's moderately bad. And so it's like turning this dial. This one goes to 11. But it's like turning this dial up and down and controlling this. And it does it at several different levels. So FoxP3 itself, and what I learned when you do this, and I wasn't a geneticist before, I've become a geneticist since, is you don't get to name your gene. There's a nomenclature committee for that. And this was the P3 member of the Fox family. So that was really boring to me. But you do get to name your protein. So we got to call it Scurfin. So that was cool. But what we also learned is that if you expressed FoxP3, the wild type, or in this case it's Scurfin, versus one that didn't have a fork head domain, that is, it couldn't bind to DNA anymore, the wild type version, you would decrease interleukin-2 production. It's a major growth factor for T cells, including regulatory T cells, but not only regulatory T cells. So you can inhibit IL-2 production dramatically. If it didn't bind to DNA, it didn't work. So that was great. So that's one way in which this thing can work. But there's also other ways. And again, you've heard this story. This is sort of the prequel to the movie. But we knew that these CD4 T cells were not very reactive in these animals. And and actually, we also learned that was also true in the humans once we started looking at IPEX patients. But we also know that this is not a homogeneous group of cells. And Shimon showed you this very picture earlier, and I think he put the red circle about exactly where I put the red circle, so I felt good about that, which is when looking at these CD4 T cells, there's a subset of them that express this CD25 marker. That is the high affinity receptor for this particular growth factor. And you now know that these cells are dependent on that growth factor, and they're dependent on expressing that particular gene. We didn't know that at the time. Shimon didn't know that when he did this experiment. But what we did was we looked at this data, and this was in 1995. And so we had cloned this gene in 1998. So this was still relatively fresh data. And we looked at this and said, okay, so what's going on there? Is that relevant? And so a phenomenally talented scientist in the group, Rolly Khatri, essentially came up with the theory that these guys must be what expresses FoxP3. And sure enough, when you look at the expression of the FoxP3 gene, it's found only in this subset and not in this subset. Okay. Now, there are ways you can induce it in this, and Sasha and Shimon have talked about that, et cetera, but in the normal homeostatic situation, these cells are the only ones that really express this particular gene, which was confusing to me because I remember I said one of the reasons I prioritized this as the last gene to sequence was that it was expressed in every tissue in the body, and that didn't make any sense, and that's still true, except if you look really closely. So when you look really closely, and this is a whole variety of hematopoietic cells on here, and you can see a few bars that stick out. These are the samples that express FoxP3, and these samples all have that subset of T cells in them. If you don't have that subset of T cells, those CD4 positive, CD25 positive cells, there's really very little expression. And the reason it actually is in all tissues in the body, is that all tissues in the body have regulatory T cells. So your muscle, your heart, your kidney, your liver, regulatory T cells are resident in all of those tissues, and they have important functions in those tissues. So if you go for a run this afternoon, you'll damage your muscle. At least I do when I go for a run. One of the things that helps repair that muscle is the fact that the regulatory T cells are embedded in that tissue, and they produce growth factors and other things. They respond to the damage, as Sasha mentioned at the end of his talk. They respond to that damage, and they produce growth factors and cytokines that help repair that muscle every time you go for a run. And so it's a way of kind of rejuvenating, and again, getting back to a homeostatic situation throughout the body. And so we were somewhat misled by the fact that the gene was in every tissue, but at very low levels, because there are very few of these cells, but they're there everywhere. So the other way that these cells work to control immunity, and again, you've heard all of this, I'll show you a little bit of our original data, is looking at the activity of these suppressor cells, regulatory T cells. And so this is the response of T cells to a particular stimulus. If you add this small subset of regulatory cells, you knock that down. If you add other cell types, in fact, you augment it. We expected that based on Shimon's data and other people's data, so that was great. When we took our transgenic mice, in those cases, all the T cells in those mice expressed FoxP3 because of the way we generated the transgene. And when we looked at them, it turned out any cell expressing FoxP3 was able to inhibit this response. And so that really told us that FoxP3 could drive a dominant process within these cells to make these cells inhibitory cells. And so that was actually tremendously exciting for us, and it really said that however this worked mechanistically, it was able to actually control a vast array of functions. And many other people, Sasha being the lead on this, have really detailed how that works at a more molecular level. So now we had some idea of what the pinnacle here was, or at least one pinnacle here was that allowed us to maintain this balance between autoimmunity and health and protection from disease. So that's terrific. So what are the therapeutic implications for this? Well, so I'll put those in three categories. One is for the patients themselves. You don't want to lose sight of that. There's not many of them, but in fact, they have a devastating disease. And so it would be nice to figure out if we can use this discovery to actually help them. The other is in autoimmunity, and clearly if these cells are important in regulating T cell responses, we should be able to use them to control autoimmunity in various ways. And you've heard a little about that. I'll show you some examples of that as well. And then in oncology. So the question becomes, are these important in rejection of tumors? Do they prevent normal immune responses from controlling tumors? And there is data around that as well. So for the patients, right now, the current therapy is to do bone marrow transplant or hematopoietic stem cell transplant and really give these patients a normal bone marrow and hence normal T cells. If you do that, they will survive, but that's not always a feasible thing depending on the patients. You can, in principle, express the FoxP3 gene in T cells that are just come out of the blood from these kids. That works. Shimon and Sasha and others have shown that that will work. That may not work as a long-term therapy because those cells probably won't live forever. They probably need some level of renewal. So the other option would be to take blood out, generate hematopoietic stem cells. We know how to do that pretty well. Express FoxP3 in those hematopoietic stem cells and put them back. And people are certainly working to progress those kind of therapies as well. Ultimately, I think the most elegant solution to this would be to take those hematopoietic stem cells, use gene editing technologies such as CRISPR-Cas9 to replace the defective FoxP3 gene in those cells with a correct version of the FoxP3 gene. Put those back in as normal regulatory T cell precursors, normal bone marrow, and put them back in. And I'm fairly confident that that will eventually work technically, but we're a little ways from being able to do that at this point. Another way to potentially do this in autoimmune patients is really to expand the cells, and there's multiple ways to do that. There's one published study I'm aware of in which T cells within autoimmune individuals are expanded using this growth factor interleukin-2. And so I said that Treg cells need interleukin-2. All T cells can actually use interleukin-2 as a way to expand and grow. And so what a number of groups have now done is they use very low doses of interleukin-2. It's actually a very toxic drug. It's used in cancer, and it makes people incredibly sick. But if you use very, very low doses, you can actually increase the number of regulatory T cells. And so the shaded bar here is patients who are on IL-2, and you can see during the course of the treatment, their number of Treg cells, and this is including FoxP3, goes up. It comes back down off the IL-2. It goes back up, comes back down, goes up, comes down. So you can increase the numbers of FoxP3 and Treg cells by doing this. There's also other strategies to use modified IL-2s that only bind to the high affinity IL-2 receptor. There are some clever protein engineering strategies that are ongoing right now that will probably be in the clinic in the next year or two. And the question is, does it have any effect on disease? And this particular study is in lupus, but there are studies in many other diseases as well. And you can see that while these people are being treated with this interleukin-2, their disease activity, and this is a lupus score, is going down. Now, it's also very large error bars on this, and it's very early days, but I think it's relatively encouraging that the idea of giving a drug that's relatively specific for regulatory T cells and can expand them can also result in disease improvement. It makes sense, and whether it will work long term, I think, is something that still needs to be determined. It's also possible to be more clever about this, and that may or may not end up being a good thing at the end of the day, but it is possible to collect blood cells, isolate the Treg cells, expand them to large numbers, and put them back in. So there are clearly clinical trials going on to do this right now in type 1 diabetes and in a couple of other settings where people are getting doses of expanded regulatory T cells and hoping that they will have a diminishment in the progression of disease. You can do a little trick here and convert this regulatory T cell and give it a different specificity. So for those of you who follow what's going on in oncology right now, CAR T cell is a very common term, and that is people putting a receptor into a T cell so that it will recognize a tumor and eliminate that tumor. So it's also possible to put a receptor into a regulatory T cell and change the specificity of that regulatory T cell. And the way you might want to do that is put a receptor in for a common antigen like insulin or GAD or thyroglobulin or MOG or factor VIII for hemophilia patients. So we know what some of these are for some of these diseases. We don't know all of them. But you can put a receptor into these regulatory T cells exogenously, grow them back up, and put them back in. And now you're putting in regulatory T cells that are highly focused on whatever disease it is you're trying to treat. And so these kind of therapies, again, they're not quite ready for prime time yet, but they certainly will be in the next couple of years. My friend and colleague David Scott refers to these as police cars, because what they'll do is they'll go in and they'll just control the offending populations of cells. This works very well in mouse models, and so we just need to translate this now into humans. I mentioned cancer, and there is a fair amount of old data suggesting that getting rid of certain populations of T cells actually improves the ability to reject cancer, which didn't make a lot of sense to a lot of people. And particularly in certain cancers, if you get rid of CD4 T cells, and these are these master cells that control a lot of immune responses, if you get rid of those, you're, in a mouse model, it's easier to reject the cancer. And that didn't fit. And I think now we would say that you're probably getting rid of the regulatory T cells. And so when you look at cancers in mice and in humans, there are a number of them in which the cancer recruits and activates regulatory T cells so that those regulatory T cells can suppress other T cells that come in to eliminate the cancer. So the question now becomes, can we get rid of those regulatory T cells in a tumor environment, but not throughout the body, because obviously throughout the body would be a problem. And so there are certainly strategies for doing that. There's a number of them. One is shown here. This is using a growth factor. It's interleukin 21, if anyone cares. But this is the growth of a tumor in an untreated mouse. If we stimulate cells with interleukin 2, so again, that is a growth factor for regulatory T cells. It's also a growth factor for other T cells. It slows down the tumor a little bit, but not very much. If we treat these animals with interleukin 21, which stimulates CD8 T cells, it stimulates cells that are good at killing cancer, but it does not stimulate regulatory T cells very well, you now see that the tumor is rejected. And so I think there are multiple ways in which we can potentially direct the immune response such that we can differentially stimulate regulatory cells versus non-regulatory cells. So again, come back to this balance. There are a lot of tools at our disposal, and I've only talked about a few of them right now, in which we think we can manipulate regulatory cells versus non-regulatory cells now that we know what they are. So I'm going to finish with two quick slides. One of which is that I said I wasn't a geneticist, but I've kind of become a geneticist, and in part it's because genetics tells you truth. When you get a gene, and if you have a mutation and you can prove it's responsible, even if you don't know the mechanism, you know you're working on the right thing, and I think that's incredibly invaluable. So it used to be that it would take a long time and cost a lot of money to sequence a genome. By next year, you'll be doing a genome for less than $500. In a couple of years, it'll be $100, which means the number of genomes sequenced is going to be huge, which means that in the human population, we're going to be able to find rare genes much more readily than we do now, and we're actually not bad at it now. We also have very powerful tools for looking at gene expression. We can look at the expression level of every gene in a single cell right now, and we can do that for thousands or tens of thousands of cells at a time. And if we can get the data sharing right, and we can link this to clinical data, we have enormous power to find rare mutations and rare effects. And I think this is going to be a tremendously valuable tool for finding therapeutics going forward. There's a very common way to look at this. That is that if a particular allele or type of gene is very common, if it's common, but the effect it has on biology is low, it's over here. They're relatively easy to find, but they don't do much. FoxP3 is an example of a gene where it's a very rare mutation in a gene, but it has a very high effect size. It has a very dramatic effect on the organism. These have been hard to find, but as we can now sequence millions and millions of people, we will have 10 million genomes sequenced in the next couple of years. It's going to be much easier to find these particular genes, and we need to be able to tie those to biology and clinical biology. And when we can do that in a bioinformatic way and get access to the data, this is going to be an incredibly powerful tool for understanding biology and for finding new therapeutic ways to treat that biology. So I'm going to stop. I don't know if I'm over time or not. I need to thank a few people. Most of this work, there's a long list of companies I actually worked at, but frankly, most of them were the same group of people. We just got bought by other companies, which is an interesting experience, so I can talk about that later if you want. But these were the people who were involved in that. These are a number of collaborators in various areas. I'm going to highlight 3 people. David Gallus, who was one of the people who really highlighted the work going on at Oak Ridge National Labs. He was the CEO of the company and really helped bring the mouse in and had the vision to work on something that frankly was not obvious for a biotechnology company to do. Mary Brunko is a geneticist extraordinaire. Frankly, FoxP3 is probably not the most important gene she discovered. There's another gene called sclerostin, which was involved in bone development, which will probably be approved as a drug later this year, and it's probably going to be more important, at least in the near term, in terms of human health, and Rolly Khatri, who did a lot of the biology. One final thank you, which is to the mice. They are not exactly willing participants in this, but they're incredibly important. And it's incredibly important we respect their contribution as well, in my opinion. So I'll stop there and take questions.
Well, do we have one burning question from the audience? I'd like to have one from the young people, from the students. Any? Is this a guy you want to work with? Yes, there is a question.
Hello.
Yes, it works.
Just now when you talk about the therapeutic, so when you do the, can you go back to the slides?
Maybe.
When you try to transplant.
Where am I going? Keep going. There? This one?
Yeah, this one. So when you say you try to edit the CAR T cell or the Treg and put it back to the human, what do you expect the thymus when this CAR Treg after being through to the thymus, whether they will be educated again to go back to the original?
Yeah, no, it's a really good question. So I think we're a couple of years away from doing this in people, so just for starters. It works in mouse. I don't think we need to re-educate, though, because we're taking we're taking Treg cells out of an individual who's already been educated, so they have the normal repertoire, et cetera. And so theoretically, that was simply insufficient to prevent the autoimmune disease, and so now we're going to add one more receptor into that. But it's a very good question in terms of what's going to happen with that cell, and as Sasha points out, if those cells are not, if those cells are plastic, that is, if they can change their phenotype and they become an effector cell instead of a regulatory cell, that would be a huge problem. So I think what you would do, I'm sure what you would do, is you would put a suicide gene in that system or something so you could kill off all those cells if it became a problem. So you'll have a safety switch in any such cell when you do that kind of approach.
Okay. Well, thank you so much. I think we should give all 3 speakers a hand for a wonderful morning.